Hello everyone. Today we will be discussing machine learning and electron microscopy image analysis, nanostructures characterization by Sophia Hale, Anderson Liu, Andrew Kim, Carter Tsao, Matthew Nadavel, Sid Mahesh, and Simon Chia. And our advisors are Dr. Starostina and Robert Downing. Firstly, electron microscopy is a technique that uses a beam of electrons to create highly detailed images of tiny objects. It provides much higher magnification and resolution than regular light microscopes. Machine learning algorithms have been primarily used to identify which data points in the image deserve greater focus. In the conversation of the consideration of automated and remote experiments, there are three main components to consider with complete automation. The engineer controls, the machine learning algorithm, and the perceived goal or target of the experiment. Engineering controls allow the operation of the microscope through external electronics, relying on manufacturer-provided programming interfaces. Next is the machine learning algorithm that generates a sequence of operations for the computer. Finally, the perceived goal or target is a list of potential outcomes based on the experiment, guiding the process. The goal of automated experiments is to minimize human intervention, as humans are more prone to errors and humans can also be inconsistent within their findings. Scanning electron microscopy is useful for analyzing small materials, but its images are difficult to process. Commonly used methods like edge detection struggle with dense or complex images. Machine learning, especially convolutional neural networks and the only look once models, can detect nanoparticles by learning from large datasets. These models are more accurate and scalable, but they need labeled data and strong computing power. While effective, their accuracy drops in dense settings. However, by combining traditional methods, machine learning, and synthetic images, detection is improving.
Here's a list of machine learning models successfully used to classify electron microscopy images. Convolutional neural networks, or CNNs, recognize images by detecting patterns like edges and textures. Cascade mask, or CNN, improves object detection by segmenting overlapping objects for higher accuracy, but is slower and more resource intensive computationally. Inception improves object recognition by using multiple filter sizes at once, capturing both small and large details, but is complex to modify. YOLO, or you only looks once, detects objects in one step, making it fast for real-time use, though it struggles with small or crowded objects. ResNet prevents information loss and keeps networks with step connections, making it powerful for image tasks, so it requires heavy computing and large data sets. AlexNet was an early breakthrough using ReLU, or rectified linear unit, and dropout, but is slower than newer models. TT, or pure geometry improvements, simple and effective stacking small filters for better learning, though it needs a lot of memory and computing power. Next slide. What are nanostructures? Nanostructures are less than 100 nanometers, have unique properties, and are useful in medicine, electronics, and material science. What's our motivation? Why do we want to image nanostructures well? Our motivation is the applications nanostructures can have for many different fields from biochemistry, quality control, semiconductors, and nanotechnology advancement with drug delivery. The imaging of nanostructures can help reveal important properties for many different applications. Next slide. There is a lot of interest in the world of nanoscience in making nanoparticles that combine several different materials, semiconductors, catalysts, magnets, electronic materials. Here's an example of a paper describing the research by a team of Penn State scientists. The team starts with simple nanorods composed of copper and sulfur. They then sequentially replace some of the copper with other metals using a process called cation exchange. By altering the reaction conditions, they can control where in the nanorod the copper is replaced. At one end of the rod, at both ends simultaneously, or in the middle. They can then repeat the process with other metals, can also be placed at precise locations within the nanorods. By performing up to 7 sequential reactions with several different metals, they can create veritable rainbow of particles. Over 65,000 different common materials are possible. Using straightforward chemistry and a mix and match modular strategy, Researchers have developed a simple approach that could produce over 65,000 different types of complex nanoparticles, each containing up to 6 different materials and 8 segments, with interfaces that could be exploited in electrical and optical applications. These rod-shaped nanoparticles are about 55 nanometers long and 20 nanometers wide. By comparison, a human hair is about 100,000 nanometers thick, and many are considered to be among the most complex ever made. Next slide.
All right, so these are our SEM datasets and their annotations. So SEM images are classified based mainly on their dominant shape and the structure of the imaged object. The dimensionality of the nanostructure, 0D, 1D, 2D, or 3D, was used as our first criterion, being the visual benchmark of the nanostructure itself. In this classification, 0D objects refer to particles which can be either dispersed, individually identified over the image area, or packed together into a clustered structure. These structures can be either entangled in bundles or mutually aligned with respect to each other. 2D structures refer to films, coatings on a surface, and include a wide range of cases differing from each other for the surface roughness. 3D structures can refer to pillars or micro devices such as microelectromechanical systems, or MEMs, typically fabricated by lithography techniques. A schematic view of this classification is illustrated in Figure 1. The dimensionality discussed above provided the main basis for this characterization, but was not exhaustive. After the investigation of SEM images, database, and discussions with SEM scientists, and additional categories such as biological samples and tips, these last, mostly related to atomic force microscopes, were introduced to enlarge the spectrum of the possible categories. The full set of selected categories is illustrated in Figure 2, with a representative image for each of them, while the breakdown of the number of images in each category is shown in Table 1. These 10 categories cover a broad range of SEM images. They are applicable to scientists working in different areas of nanoscience. At the same time, they're chosen as more distinct from one another as possible. Next slide, please. All right, so our starting point. So far for this project, we have written our code partly with the help of ChatGPT. We are incorporating OpenCV, which is a computer vision library. We're applying it to 18,500 SEM images, which we have split into 5 main groups. The first is spherical particles, which you can see up in the top right. Then we have cubical particles, shown in the right center. Beyond these, we also have patterned surfaces, nanowires, and pores. Each of these categories has its own unique features. If you want to get a quick idea of what each category looks like, you can check out our legend on the slide. Next slide, please. All right, so our data sets and our preparation strategy. To prepare our data sets, we plan by splitting the data using a 70-30 ratio, which is an industry standard, between in-sample training data and out-of-sample validation data. For the training set, we'll experiment with increasing proportion of data using a scaling factor of 10. We will label the training data using image processing techniques to enable machine learning. The model will be trained on different subsets of data, and we'll evaluate and record its performance using the same training data. Finally, we'll visualize the results through performance plots. Our expected outcomes are nanoparticles count, the morphology table, the outputs, and the ability to read the scale bar from the bottom of any SEM image. Next slide, please. All right, so these are our results for spherical particles and the learning curve for our model. On the image to the left, there are 217 particles in the image. And with a series of 5 trials using the number of particles incorrectly identified, it was used to construct a learning curve. For trial 2, shown in the top corner, it failed to correctly identify 129 different particles because it failed to distinguish between individual particles and clusters of particles, which compared to trial 5, which is shown in the bottom corner, right corner, which incorrectly identified only 24 particles. It shows an improvement, as it was much better at distinguishing between individual particles and particles in a group cluster. This gradual improvement can also be shown by the red regression line in the graph, showing the trend of our data over time. Thank you.
So these are our results for the cuboidal model. As Matthew spoke about, we have several different types of data sets. This covers the cuboidal particle data set. As you could see on the left, this is a graph of different contour values versus against the shape detection and accuracy. The shape detection and accuracy being on the y-axis and the contour values being on the x-axis. What contour values control are how sensitive the model is to the defining factors of a particle or structure, that being like boundaries and edges and whatnot. So as you can see, we tested 5 different values, 0.01, 0.02, 0.04, 0.08, and 0.16. As you can see on the graph, we had a sweet spot and our lowest shape detection and accuracy at 0.04. You could also see what that looked like from like output from our model on the right. It is the picture farthest to the right and at the bottom, also highlighted in green. Continuing that, our model has also been able to output what it identifies in a table format, the morphology, the area, the effective radius, and the total particles detected. The morphology being the shape, so like triangle, rectangle, pentagon, square, circle, and if none of those, blob or unknown. And for the area and effective radius, both of them are calculated in pixels. So in the future, our next steps are to be able to read off the scale bar at the bottom of the SEM images. Currently, the AI cognizes it and completely avoids it. So anything behind the scale bar, any particles behind the scale bar are not included. It just completely ignores it. So our next step will be to read off the scale bar, the relative size, and then from there calculate the relative size of the particles according to the scale bar. So just going over everything for the cuboidal model, it has reached a detection accuracy of 90%, that being the particles count, morphology, recognition, the shapes I spoke about earlier, the area and effective radius, both in pixels, and avoiding the scale bar. You could see on the right a clearer picture of the output of the model, highlighting the particles that it identifies in green and labeling them with what shape it is.
Okay, as for future work, our first step is to ensure the system can reliably read the main scanning parameters directly from any SEM machine. Right now, a lot of these parameters have to be entered manually, which can slow things down or introduce errors. By automating this process, we'll make the analysis more consistent and adaptable. Next, we want the system to measure the real size of the particles, how they line up, and what kinds of patterns they form. This will make the results more clear and more accurate. Finally, the ultimate goal is to integrate our code directly into the SEM operations software. If we achieve this, the microscope will not only capture high-quality images on its own, but also analyze them in real time. This would make the SEM analysis faster, more accurate, and much more scalable for future research. Next slide. Here are our references. Next slide. We would like to give a sincere thank you to our parents for the support they have shown us in our journey, as well as ASDRP for allowing us to conduct this research. We would also like to give a big thank you to our advisors, Dr. Starostina and Professor Downing, for guiding us through this research process. Any questions?